In this lecture, what we're going to do is we're going to look at what happens to finish the cellular respiration in the presence of oxygen. Cellular respiration started with the reaction of glycolysis. Glycolysis occurs in the cytoplasm without oxygen being present. During glycolysis, what gets made are 2 molecules of pyruvic acid and 2 net ATP. Also, we have the 2 molecules of pyruvic acid. Now, if oxygen is present, those 2 molecules of pyruvic acid will move into the mitochondria because oxygen was present. By the end of the Krebs cycle, what I should say is by the end of the bridge reactions and the Krebs cycle, what is produced are 4 molecules of carbon dioxide, 2 more molecules of ATP, 6 molecules of NADH, and 2 molecules of FADH2. Now, we're not going to spend a lot of time on these. What I do want you to know about them is simply that they are carrying electrons, okay, in the form of the hydrogen. Okay, so electrons will then be used from these hydrogens in the electron transport chain. And that's the last little bit that we need to look at in making ATP in the presence of oxygen. So we already had that slide. We had this slide yesterday, so this is the first new slide. It says the electron transport chain. The electron transport chain consists of a series of proteins that are embedded in the inner membranes of the cristae. That's those like lazy river looking things that are inside the mitochondria. And mitochondria in the eukaryotic cells. Okay? In prokaryotic cells, the electron transport chain lies along the cell membrane. We're not really focusing on prokaryotic cells, we're mostly focusing on eukaryotic cells. In this last stage of aerobic respiration, which simply means in the presence of oxygen, NADH and FADH2 will release the hydrogen atoms, generating energy to produce the ATP. What is the total number of NADH and FADH2 that has been produced so far? Okay, well, we are not going to memorize these numbers, but it says that there are 10 NADH and there are 2 FADH2. So here we have a total of 10 hydrogens that are going to be available to the electron transport chain. Here, 1 FADH2 has 2 hydrogens, so 2 of them have 4 hydrogens. So altogether, we have 14 hydrogens that are available for the electron transport chain. The purpose of NADH and FADH2 is to carry these high-energy electrons into the electron transport chain. The electron transport chain uses the high-energy electrons to convert ADP to ATP. So in other words, the energy is going to be stored in the bond as an extra phosphate group gets added to ADP to become ATP. So, steps of the electron transport chain. First, some of the little bits are going to be added to the diagram. So here we have our NADH. We've got our 10 that are available, right? Electrons are moving through the electron transport chain. Here's the FADH2. It only states 1 molecule right here. So 2 hydrogens are being released to become FAD+. It says the high energy electrons from NADH and FADH2 are passed along the electron transport chain from one protein to another. So here are the proteins right here. At the end of the electron transport chain, the electrons and the hydrogen, right, because the hydrogens were being released from both of these, will be combined with oxygen to form H2O. And we've got that little bit being illustrated right here. Oxygen is the final electron acceptor. Oxygen is essential for getting rid of the low energy electrons and the hydrogen ions. As these electrons move down the electron transport chain, they release energy. The energy is used to pump hydrogen protons, right, the hydrogen ion right there, across the membrane from the matrix to the inner membrane space. The hydrogen protons are pumped against the concentration gradient from an area of low concentration in the matrix to an area of high concentration in the inner membrane space. So in other words, ATP is being used to move things against the concentration gradient. That was an example of active transport. A concentration gradient has now been established. There is a high concentration of hydrogen in the inner membrane space and a low concentration in the matrix. Also embedded in the mitochondrial membranes are enzymes called ATP synthases. Hydrogen ions flow through the ATP synthase back to the matrix and the low area of low concentration. As the hydrogen ions flow through ATP synthase, it spins a rotor. Each time it rotates, a phosphate is attached to ADP to become ATP. Okay, so that's a lot of detail. This is the process, so you could study it further if you want to understand more about the electron transport chain. I'm not going to be asking you like how many NADHs, how many FADH2s. The big thing I want you to know from this, okay, is there's a couple of things. One, you need to know that the Krebs cycle is not the end of the reaction, right? You need to know that the last step in the reaction is the electron transport chain. You also need to know that the electron transport chain is what produces the massive amount of ATP that we are able to obtain from one molecule of glucose. You also need to know that the electron transport chain does occur in the presence of oxygen. All right, recap of the electron transport chain. The system couples the movement of high energy electrons with the production of ATP. As the high energy electrons move down the electron transport chain, they release energy. This energy is used to move hydrogen protons across the membrane. These ions then rush back across the membrane, producing enough force to spin the ATP synthase and generate enormous amounts of ATP. So let's look at this part. Actually, it's the next slide that we really want to look at, but this is part of it. Let's summarize what has happened prior to getting to the electron transport chain. So there was glycolysis, right? And what glycolysis will produce is a net gain of 2 ATP and the 2 pyruvic acids. Then there was the bridge reaction, which we're really not going to focus on. The bridge reaction, the big thing is that it produces some of these electron carriers, right, in the form of a hydrogen ion. Then there's the Krebs cycle, which produces 2 ATP, 6 NADH, and 2 FADH2. All I really want you to focus on is that it produces carbon dioxide. Remember that there was carbon dioxide produced here, and more ATP. So glycolysis, so you probably need to write this down extra from what you just see on these notes. Glycolysis produces the 2 ATP and the 2 pyruvic acids. The bridge reaction produces, I believe it was 1 carbon dioxide and this electron carrier. The Krebs cycle produces more carbon dioxide and more ATP. And then, okay, but here's the big thing. Each NADH has enough energy to produce 3 ATP. Each FADH2 has enough to produce 2 ATP. So we have that. We have the numbers from the Krebs cycle all together. And here's what I would like you to know. And not even the exact number, just the idea that because of the electron transport chain that occurs in the mitochondria in the presence of oxygen, one molecule of glucose ultimately will produce 38 molecules of ATP. 38 molecules of ATP. So you can see glycolysis makes a little bit of ATP, but it's not particularly efficient because it's not able to really get the energy that is stored in the bonds of the glucose. The Krebs cycle also not particularly efficient because it makes a little ATP, but that's just it. It makes a little. The electron transport chain makes 34 ATP. Much more efficient than the glycolysis or the Krebs cycle. Okay, and all together, if oxygen is present, the glucose will go through glycolysis, will go through the Krebs cycle, will go through the electron transport chain, and make 38 ATP. It goes on to say only about 40% of the energy contained in the glucose molecule, and don't forget we're really talking about the bonds between the atoms, has been converted to ATP. The other remaining 60% is given off as heat. So, you've got glucose. It starts to be transformed so that ATP is produced. The first step is glycolysis. Glycolysis occurs in the cytoplasm without oxygen. It's anaerobic. If oxygen is available, then the 2 pyruvic acids will enter the Krebs cycle. Since there is still oxygen present, after the Krebs cycle, you have those electron carriers that will move the electrons into the electron transport chain and produce an additional 34 ATP just by the electron transport chain. So if oxygen is present, a total of 38 ATP are made. What if there is no oxygen? What if the glucose starts getting broken down by glycolysis, makes the ATP, and there's no oxygen available? That is what the last lecture will be on tomorrow. The last lecture will be on what is called fermentation.